was like, you know what? I'm just going to go. 90%, and then I had like this one set of data that was good.
Yeah.
And I'll never forget it.
So maybe it's good.
And he was like, has anyone ever shown that before? And I was like, not that I know of. I always assumed that I had done something wrong because I was a very unconfident PhD student. And I'm like, I don't know, Ben, like I'm dumb. So I did think to myself, maybe my cells don't like the light. Only the trained eye will recognize me an idiot or incompetent or stupid or anything.
It's tough.
I think I would have shut down completely, and then I would have really thought, I don't know, maybe I shouldn't be doing this anymore. Why didn't I put that? I don't like doing like R&D with the pressure of all this capitalism. The antagonists are the biomolecules themselves. Oh my gosh!
So this is So Blake, welcome back to Baltimore. It's nice to see you in person.
It's good to see you too. Thank you for inviting me.
How does it feel like being back at Hopkins?
It feels like I have come home. Honestly, like the vibes are just, I miss them so much. Like I just saw my boss in the hallway.
Yeah.
I took a Zoom meeting in Rangos, and
I know you're about to meet a lot more people.
I'm gonna go visit my old lab. I haven't even been there yet. Yeah. I've seen like a few changes, like, oh, there's a parking lot there now.
Yeah, something different. Right?
Yeah. Like,
must be nice. I think it's always good to come back. So since we are here, could you just give us a quick sense of your overall journey, like what you did during your PhD and what you are working right now?
Yeah. So my original background is in biomedical engineering from undergrad, which I did at University of Cincinnati. And then I pivoted to molecular biophysics for my PhD. That program had we had to do at least 3 rotations. I did 4. My 4th rotation was with Bin Wu, who is a single molecule RNA researcher. And that was definitely not what I expected to be doing at all. But I think, again, I think it was vibes-based. Like, I felt the good vibes from him and his lab. Yeah. He was a new PI, and I was like, he was very energetic and passionate and very nice. So I was like,
genius
I think this could be he's a genius person. Yeah. I was like, I think this is going to be a good lab for me to be in. And so it ended up being, my PhD was all about studying like RNA decay and how the dynamics and the kinetics of it and things like that. I ended up, finding this tiny little organelle that I proposed. I mean, it had already been found before, but its function wasn't very well known. But my project ended up revealing that this tiny organelle called P-bodies was responsible for fast RNA decay whenever fast RNA decay is needed.
Yes.
And so I was, really excited about that finding. It gave me an opportunity to publish, my first author primary research article. I published reviews and perspective papers, methods papers, stuff like that. Gave a lot of talks too at some big conferences, and that's the main thing I got out of the PhD, because I from my actual PhD research, I'm not doing anything with RNA anymore. I actually, during my PhD, I started becoming more passionate about sustainability and also removing plastics from the environment, removing animal-based materials from the environment. I started the Johns Hopkins Alt Protein Project, which was focused on making food alternatives. That led me to this whole cycle.
To your postdoc.
To my postdoc, where I'm now in David Kaplan's lab as a postdoc. And he's a cultured meat researcher, but I'm not doing cultured meat. He's also a biomaterials researcher. So he's a leader in bioplastics, as well as just biomaterials for tissue engineering applications. And he also has an interest in textiles and just biomaterials in general.
So that's what you are focusing on right now.
That's what I'm doing now, yeah. Specifically, I've been trying to develop an elastic alternative that is biodegradable and compostable, but renewable resources. That's been extremely challenging, but
pretty cool. We'll get a bit later on that. So it's quite a good overview. So since you're here, maybe let's reminisce on your PhD life first. So you mentioned like choosing your PhD lab was kind of just a vibe thing, right? And remember you told me you just applied 2 PhD programs.
Oh, that's a good memory. Yeah.
Was that so? Could you tell us like why did you choose to do a PhD and how did you end up like choosing biophysics?
Yeah, good question. So yeah, I did only decide to apply to 2 universities, and I I did the same thing for undergrad too, by the way. So this is just like a theme. I'm very decisive, and
very
also
focused.
I just don't really like spending that much time on applications. Like
that's fair.
I'm not saying it's a good thing,
it's wasting time.
I applied to UC Berkeley and Johns Hopkins, and I got invited for interviews to both, but did not get into UC Berkeley. But I did get invited to Hopkins.
That makes things easy.
So I was like, and I did have a backup plan though. I wasn't sure if after my undergrad, if I really wanted to go to grad school or not.
Right.
But someone gave me the advice that, okay, you don't have to go to grad school. Why don't you just do the GRE? Because at the time we had to do that. I don't think they require it anymore.
Now I think they no longer,
Yeah, they were like, just study for it, get it out of the way, get all of your letters of recommendation in a row. Like, get all of that together. Because, the longer you're away, people will start to forget about you. So get those letters of rec.
Yeah.
Do the application. Do the interview even, like, if you're invited. And then...
And just try it out.
See if you just see what happens. At least you'll have, at the very least, you'll have all of your grad school application materials ready, and then you can apply the next year. I thought I applied for Fulbright. I really wanted to do an international thing. I thought
that's
about
a cool one.
I thought about industry, but then I got into Hopkins, and I was really excited about it, and I was like, you know what? I don't even need a gap year. I'm just gonna go.
Nice. Yeah, I think that's normal. I think usually like people before doing PhD, I don't think they have a full picture of what PhD really is. Right? Usually we just fight our way out. So when you first started, did you have any imagination of like what your PhD would be like, if like you put yourself in the shoes of you maybe 6-7 years ago?
I definitely did. And you also asked me, I realized I didn't answer it, you asked me why biophysics.
Right.
And part of the reason what launched me into biophysics was I just found out that I really, really liked understanding how things work on a very, very fundamental level.
Mechanistic.
At the molecular level. I really appreciated that. I actually did, when I was an undergrad, we had to do an internship program. We had to do, it was at the University of Cincinnati, so it was called a co-op program. We had to do at least 5 internships, paid internships in industry or academia. And
That's a really good opportunity.
It was amazing. Like my first 2 were in Procter & Gamble when I was like a sophomore. And they paid really well. They treated me really nicely. I was in a modeling and simulation group, and there were folks at Procter & Gamble who were modeling, like the skin molecules and how a lotion would, interact to like open or close a pore. And I remember they were like modeling the pore, and I was like, oh my gosh, wow, like
so cool.
That's and so it was helping with understand like designing better cosmetics in their case, and also developing some of their claims. I also did a project where I was modeling the human sinuses to see if their product Sinex actually removed mucus or not, and I think it turned out that it did, and we published it. So I got just really excited about just understanding things on the molecular level. And then it started launching my interest in biophysics because that is really just like the fundamental thing that makes cells and biomolecules
that makes sense.
Work.
I see. So I guess, okay, that's kind of what you would imagine, like, doing your PhD, like what your PhD would be like.
I mean, I did think that I was going to study medicine in some way. I thought I was going to study like how drugs and come in through cellular pores to have downstream effects that are favorable. I thought I was interested in therapeutics.
How drugs are delivered into the cells.
Exactly.
Yeah, that's cool as well.
I was interested in that, but then I just think you should enter your PhD with some goal, but also just realize that it's probably going to change.
Yeah.
And I joined some membrane biophysics labs at first, and it wasn't speaking to me in some way.
Sometimes it's a vibe, everything matters, right? The people, the PI, right?
Right.
It could be. So when you first started, when you really got into the lab, starting your work, did anything surprise you in a way, or just maybe like, oh, that's not what I imagined, or that's something unexpected? Is there anything?
Everything surprised me. I was such a fish out of water, even though I had all these internships and research opportunities, undergraduate research in my undergrad. Coming to Hopkins, coming to Bin's lab.
Still different.
It was so different. I just I was learning all this these more biomolecular techniques. Previously, I had done more bioinformatics and some coding type stuff. And I guess one thing that surprised me is that I think at the time I was just really impressed by how people came up with new ideas. At the time I was like, oh gosh, I cannot even imagine coming up with a new novel idea. Like, how do people...
Because you were not, I guess, used to that, the thinking in that field.
I was not. And that's why I'm so glad I did my PhD, because, I mean, I guess you can maybe get those skills of coming up with novel ideas and figuring out how to test them in other industries and whatnot, but, I think that was one of my biggest trainings from the Wu Lab was, just, I don't know, I would just have, these stupid ideas, and they were really bad and didn't make sense. They weren't feasible, and I would be like, how do people do this? And then, with enough time and enough reading papers and with enough being shut down by my PI because my ideas made no sense, I finally started to synthesize what's actually possible, and what's, I think, just the feasibility of it all.
And coming up with some good ideas yourself.
Now I come up with good ideas all the time. Actually, no, I don't. They're actually still bad.
Nah.
But they're not as bad.
Yeah, that's a fair way of saying it.
And I think before I could barely come up with ideas, and when I did, they were bad. Now I come up with all kinds of ideas all the time, and I am able to test them and de-risk them in the lab really quickly.
You know how to test, how to
I know how to eliminate.
I feel those parts are also more valuable, right? How do you test your ideas, right? That's what PhD kind of taught us.
Yeah. And then also I'm really happy when I realized I used to be oh darn, my idea didn't work. But now I'm a lot more excited when something doesn't work, because I'm okay, I need to eliminate that as soon as possible.
Yeah.
I think 1% of my ideas are actually good. So it's really a numbers game.
Yeah. I think for all scientists, I think 1% is pretty high.
Yeah.
And if your data is 1% positive, that's pretty good.
I know.
Or we should dwell on that and just enjoy that part.
My PhD was 90%. And then I had this one set of data that was good, and I'll never forget it. I used that data like I floated on that data for like the rest of my PhD, but like no, I had a whole paper that I based off of that data. But you know, one day you will get some nice data.
Yeah.
That.
So actually, I printed out your paper. So maybe it's good to refresh some memories.
No way.
So I'm sure looking at this, you'll have way more stories in your mind than me.
I have so many stories because so much of this was like accidental findings, you know, that ended up meaning something really influential in the field.
So you alluded to it a little bit about this fast iron decay system that you developed. I don't think we will get too much science here, but maybe just can you still remember one day that you feel great, and it's about this paper, maybe one day of experiment that you still vividly remember?
Yeah. There's like so what I was just describing to you, the first day I got good data, it was this flow cytometry graph.
Oh, this
figure 1B.
Yes.
I remember like the specific day. I remember there was someone walking by and I was like, "Oh my god, look. The black part is high and the blue part is low."
Yeah.
And he was like, "Oh, how'd you do that?" I was like, "I added a drug and it like caused the RNA to decay, which caused the protein to decay in this flow cytometry graph." And he was like, "Has anyone ever shown that before?" And I was like, "Not that I know of." And he's like, "High five." And then he moved along.
Oh my gosh.
It was Dan from the EWAL lab. But like this data, like this changed the trajectory of my PhD because I finally showed that I could control protein expression of the specific thing, and also that it was specific to my gene that I was trying to control.
The drug you added worked.
It was a rapamycin.
The system you designed, FKBP.
Yeah,
FRB,
and then I verified it in the RNA. But what was I think also the most exciting thing was there was another one. It was when I saw these little particles form. They were the processing bodies, but I didn't know that at the time, the P bodies. I saw these random puncta. I applied the same system to like an endogenous gene that we had, where I could knock down the RNA controllably. And then I saw, when I did this, there were these huge clumps, like huge granules.
Right.
And in my head, I'm just thinking, because I always assumed that I had done something wrong because I was a very unconfident PhD student. If I ever saw anything, I would just be like, "Oh my god, I probably messed something up." And like, I saw these puncta, and I was just like, "Oh, those are supposed to be RNA, but probably like my probe aggregated.
Why is it so big?
Why is it so big? It's probably because my probe aggregated, or maybe I had the wrong temperature, or maybe I didn't have the right imaging settings."
I see.
And so I would show this in my update for, I didn't even say anything about the granules, but then Ben asks, he's like, "Why are these granules so big?" And I'm like, "I don't know, Ben, like I'm dumb." Like,
did he know? Did he know at that point?
No, he did not.
Wow.
And then Yang, the postdoc that I worked with, he was also in that meeting. He was like, "That's weird. You should repeat this and see if those like granules still show up." And I was like, "Yeah, I don't know. I probably like just need to turn down the intensity of the laser or something. I don't know."
Right.
He was like, "Yeah, but you only see it in this sample." And I was like, "Okay, yeah, I'll just test it again." And I kept seeing these granules every time. And finally, my boss was like, you need to do an immunofluorescence to figure out what's in these. So I did some P-body markers and some stress granule markers, and there were P-body markers that co-localized.
That's very cool.
So.
I didn't know that. I thought you guys were originally trying to study P-body function.
No, it was an accident.
That's so fun.
And then what I was confused about was, well, all the literature says P-bodies are not related to RNA decay because we've never seen RNA decay there. And so I think in my last experiment, I was really excited to show that the reason that when you go to image this, just by the act of imaging and observing it, you see different
stress
behavior.
Remember, you tested different conditions, right, of laser bleaching, and some have more stress, then things don't work very well.
Yes. That was another thing. I thought that I was messing up my live cell experiments. I'm like, oh my God, Ben, I'm so sorry. I can't repeat this data. And I'm blaming myself. When really, it's because when you image the data, or when you image the cells, they get stressed because they're alive. And when they get stressed, they hold on to the RNA a lot more, and they do not decay it.
How did you find this solution or explanation from blaming yourself to eventually figuring out that's really happened?
Yeah, I mean, I think...
Is it been maybe or some like lab meeting?
I think I eventually, I repeated this so many times, and I was like, I think I probably literally cried at some point. I'm like, gosh, why isn't my data repeatable? Why are my live cell experiments different from my fixed cell experiments? And I started to, I don't remember exactly why I thought they could be stressed, but I did one day, I was like, let me just try lowering the laser intensity, because maybe I'm like, because I have seen my cells, they run away. This in the live cell, they run away from the light. And
that's such a good hint, huh?
So I was like, maybe and so it sucks because after 2 hours I go back and my cell is no longer in the frame. So I did think to myself, maybe my cells don't like the light and they are stressed out by it.
That's a genius.
I lowered the intensity, and then finally I saw the RNA entered the P-bodies.
Yeah.
And then it went away. Before, it was not doing that. It was entering the P-bodies and staying there. And then that made sense why so many papers in the field, they always said, no, we looked at single RNAs.
P-body just for storage.
Yeah.
It's not doing
just
anything.
For storage. We've never seen RNA decay there. But then why is there so much RNA decay machinery, huh? And then finally, my paper showed that when you stress out a cell through like arsenite or light stress, or I didn't test this, but maybe temperature or like temperature changes, the RNA stops decaying.
Yeah.
Maybe whatever was already decaying finishes, but by the time you collect the P-bodies, the RNA has been decayed. Or it has stopped decaying for the transcripts that are left. And yeah, I think that's a good example of how a lot of scientific discoveries are based on accidental observation. And so, but only the trained eye will recognize when something is actually meaningful.
Yes.
I did not have that trained eye before, but Bin and Yang, the postdoc that I was working with, they did have the trained eye for that. Luckily I was working with them and sharing this. Now I have the trained eye.
That's good. I think you also have perseverance that at least keeps you there, right? And then you can, and you're willing to test more times or conditions, right?
Yeah.
And eventually you could find it, right? Yeah.
Right. And I think that I give myself a hard time for why was I so insecure about my data. I think it's that I always blamed myself for if the data didn't make sense.
It's complicated experiments, right? Like it's easy to make mistakes.
Yeah, but like I was doing them right in the end. There was like some new phenomena.
You mentioned a lot of it's good. You mentioned a lot of the ups and downs. Was there a time that you felt like, oh, I don't want to do this anymore, or even thinking about quitting? Is there any time, or you always like just trying to solve the problem and then just go ahead?
Yeah.
Was there any like darkest moment in your PhD?
I guess definitely the COVID times were kind of weird. And I also, just being, in my 20s, had a lot of personal issues that I was navigating in my relationships and in my health, my mental health. And so definitely the pandemic culminated with a lot of that, and exacerbated some of those things. But I definitely never was like, oh my gosh, I need to quit. I think because my PI, Ben, was always very understanding, and he was also very patient with me, which is pretty miraculous given that he was like a new PI and he really relied on this data for his career. I think that made it so that I never felt like I need to go.
Yeah. I think PI matters a lot.
Yeah, if my PI, I've heard of some people with bad PI relationships that they kind of put them down.
It's really hard to survive on your own, if you are facing those comments or those.
I can't even imagine if Ben had ever called me an idiot or incompetent or stupid or anything.
It's tough.
I think I would have shut down completely, and then I would have really thought, I don't know, maybe I shouldn't be doing this anymore.
Right.
But I always had a very supportive lab environment, and that's why it's so important to join a lab based off of the vibes and not the science.
Because you're staying for 5-6 years there. At first, you should care about your mental health.
Right. If the vibes are bad, then you need... You're going to suffer. Yeah. Even if you're doing the exact topic that you want to be studying.
But doing science is hard. There'll be a lot of setbacks. So it's really important to choose a good lab.
Right.
Yeah. Was there any experiments that you did a lot of times, tried a lot of times, but didn't end up in the paper, and you feel like, oh, I wasted so much time? Was there anything like that?
Oh yeah, there were so many projects that did not end up here. There was a whole other project that I had going on. This is this uses chemically inducible dimerization, CID systems.
Rapamycin induced.
Exactly. But then there was a optogenetic system as well that I developed at the beginning. I actually did the optogenetic system before I did the chemigenetic system.
Like the Cry2.
It was, Cry2 CIDN. Those were optogenetic proteins from, what's it called? Arabidopsis thaliana.
Right.
Arabidopsis thaliana.
Yes, something like that.
It's a plant. So plants naturally have these optogenetic proteins because they're photosynthetic. We had these proteins that were from the plant. They dimerize under blue light. And I did lots of experiments with those.
Yeah. How did it go?
So they work. It's just not as robust. So the binding of the FRB, FKBP in the presence of rapamycin is strong. And all you need to do is add the rapamycin once. So it's a very robust system. With the light, you have to optimize how much what intensity of light, because you don't want to stress out the cells either.
I see, yeah. Light has some double-sworded effect.
Yeah.
I see. Yeah. So that's pretty cool. So after all this work, did it affect your perspective for what you'll be doing in the future? When did you start thinking about life after PhD?
I probably started thinking about life after PhD not for a really, really long time into my PhD, which is later than you should. But I definitely think that co-founding the Alternative Protein Project here at Johns Hopkins gave me a much greater feeling for what I want to do with my life. And
how did you start getting into this Alternative Protein Society? Was this somebody invited you or something just inspired you?
Yeah, I mean, during the pandemic, I had a lot of free time on my hands to explore new industries that I hadn't really thought of. I did start learning about this work in cultured meat. I found out about this because I've been a vegetarian since I was 11 years old, and then I was also extremely militant vegan at the time. I'm a little bit more chill vegan now, and but I've always been motivated by removing animals from my diet and from my clothing.
I see.
And I would think to myself, is there any relation between what I'm doing now to these
translatable kind of
to these
impact
industries? Yeah, exactly. And so I would think about, could my chemically inducible dimerization or my optogenetic dimerization...
That'd be pretty cool.
Yeah, like, could it do anything, like, with alternative foods or alternative materials? And I started getting really obsessed with the idea of using optogenetics for patterning, for patterning fine shapes. I really...
Yeah, they can do that, like tightly control the patterns.
Yeah, so it was like, I can shine lasers at my cells and then make them do stuff.
Yeah.
So I'm like, what stuff could that be for materials?
Mm-hmm, mm-hmm.
And yeah, I started realizing food. I love the food industry. I'm happy just eating tofu. So I guess. But I was becoming increasingly frustrated that there's not very many good alternatives for leather or feathers or just like clothing in general, like stretchy clothing. It's either, all the vegan alternatives are plastic. And that's not good either, because that's not good for our ecosystems. It's not renewable. It's not recyclable.
So there gotta be a better material.
Yeah, so I'm starting to feel more pulled towards the textile industry and the biomaterials industry, because it was more like for selfish reasons. I'm like, I want a cute leather jacket that's not made from animals or plastic.
That's fair. And that field is underdeveloped, right?
Yeah,
so
it's not very well.
It'd be more exciting to, maybe have some good discoveries, good inventions.
Yeah, so I started thinking about, proteins. How can we get proteins to be better biomaterials? I thought about optogenetics. Nowadays, I am like deep into bacterial cellulose work at Tufts University. I've been deep into solution fiber spinning, but what I'm realizing is that there's a reason that these biomaterials are not out there yet, and it's because they are very, very, very difficult to work with.
Is it the production or just, what's the part that is difficult?
Yeah, it's the production, for sure. It's because, I could go into so many details about this, but plastic polymers, they are able to be melted. And the fact that biopolymers cannot be melted, they just dry and they become brittle.
I see.
They cannot be processed in the same way. And then also biopolymers are very heterogeneous. And so you don't have as much uniformity. So that's another issue. The other thing is biopolymers are mostly used to being in a hydrated environment. So I was really hoping that I could use like a recombinant elastin protein, like elastin in our skin.
But they must be in an aqueous environment.
Right, they're elastic.
But not like what we're putting on every day.
Exactly. I don't want wet clothes.
That would be interesting.
Yeah, so that's why there's so many challenges. But I'm still in it.
Yeah.
I'm still in it. I'm not going to give up. I've only been in this field for 2 years. And, if I have to change direction, change labs, change funding sources, whatever, I really am passionate about making a difference in biomaterials. And I remember from my PhD that I do believe that the accidents will come. Accidents will come.
Actually, that's what I want to ask. Like, since you are working with kind of very different things from your PhD, right? Have you found anything you learned in your PhD that unexpectedly are helpful right now, like for what you are doing?
I think one of the greatest things I learned in my PhD is just how to communicate science to other people.
Especially for translatable research.
Yeah.
Right? Or maybe for funding purposes.
Right. I think I've definitely learned. I mean, in our PMB program, we had a heavy emphasis on presentations and proper presentation technique. There are a couple PIs in our program who are just very, very strict about science communication. Karen Fleming is one of them, and I was one of her rotation students in my first year. And she actually has a huge list of rules that she uses for presentations.
Yeah.
And those, I still use all, if not most, of those rules in my day-to-day life. So I'm very thankful for the presentation skills that, and Ben was also he would always emphasize, you must tell a story. Scientific storytelling is so critical. So many people go up to a presentation and they give a book report. They're like, I did this, and then also I did this, and then also look how smart I am. I also did this, and then I did
all
this,
in chronologic order.
It's like that's not how we give scientific talks. That's how you can give your papers, your boring papers.
Have you? Yeah, talking about this paper. You mentioned sometimes just changing how you tell the story makes a big difference. Did you ever have a moment where maybe hear how you frame this story actually just suddenly made the whole project just click or just be better?
Yeah, that's a good point. I mentor several students at Tufts now, and I always tell them, you do not need to tell the story, that your scientific presentation story in a chronological order, because sometimes there might have been insights that came later that informed earlier experiments, and you don't have to, unless
you're the god, you could predict everything before you do, right? Otherwise,
right. Definitely in this paper, it's so funny that you printed this out. I mean, definitely I did not present these in the most chronological order.
I think even reverse.
Yeah, so definitely I know that I did the endogenous genes way earlier than I presented them.
Because they're more helpful for demonstrating.
So Ben and I would go back and forth all the time, should we talk about the immunofluorescence assays that we did?
Yeah.
Before or after we talk about the stress experiments, and we would do that all the time. I mean, I think what's most important is that things flow in your paper and also in your presentations. They flow in a way that feels good, that's it's clicking. And I was happy because I told a story in my thesis. I had art in it. I actually asked our undergrad Marie if she could draw some artwork for to illustrate what was happening in the processing bodies. I had this whole analogy that processing bodies are like an RNA salon where they do little haircuts and massages on the
it's a little I'm not sure. I don't think this is the one you
That's
mean.
not what I'm talking about.
It's too scientific.
Oh, I wish I still have the art. She drew little RNAs that were getting their hair cut or had really long hair or were in a line at the salon. I related all of these things to the biological phenomena that I saw.
That's so cool.
And you know what? My mom, I think my mom, who was at my thesis defense, she asked the best question at my thesis defense.
So when you talk to, outsiders, definitely it's helpful to design how you illustrate the story, right?
Right. Yeah. I was just glad that she didn't get lost. She didn't get completely lost.
Then she's super smart.
Yeah, my mom, she's in government, in the bank. She's in banking, and she was like, Lauren, can you explain what the consequences are of the RNA decay rate being faster in the P bodies compared to the cytoplasm?
That's an amazing question.
I was like, Why didn't I put that? Why would I? Why would I just share these results and then not even explain what the
Sometimes we focus too much because there's so many details, so many stuff. We even forget the biggest impact, right? The biggest consequences.
What an important question.
Yeah.
And then people clapped for her.
She deserved it for sure. Yeah, we can put your artwork in this video if.
Marie would love that. Yeah, Marie's actually in BCMB right now.
I see. And then also the rules for presentation. I think maybe many trainees will need that.
Oh, Karen Fleming would love if that were shared more widely.
I know you're also doing some kind of guiding work on presentation, right? Helping others.
So Karen had a list, like a document, and she gave it to all the PMB students. And I made a presentation form of that list that I gave to all the undergrads that were participating in our summer research program in the Kaplan Lab. Mm-hmm. And so I can share either of those or both. Yeah. But it's so important. My undergrads have told me that the way that I've encouraged them to present, which is based on the slide rules that Karen had, it really transformed the way that they present, and it transformed the way I present too. So I just find it to be like one example is they say don't overcrowd your slides, one figure, literally one.
Nobody can pay attention to everything, all the distractions. One
figure one picture per slide with a title that doesn't just say mechanical properties, something like that. It'll be like the tensile strength of the treated sample, a statement, a declarative statement. You want to have the point of the slide because you have to assume that people are going to doze off during your talk. They're going to fall asleep. They're going to be like, oh, I gotta check my email. Oh, what time is it? Oh,
I did that all the time.
And then you need them to be able to look up and know exactly what you were talking about from the title of your slide. Methods, titles. You need to say, we used circular dichroism to determine the secondary structure of this protein. I don't know, something like that.
Just somebody jump into your presentation, they can already follow you.
Anyone who's late, they'll be like, oh, gotcha.
Yeah.
And there's a million other tips like that, but I think keeping it simple, not overloading your slides, and having a declarative sentence at the top, those are the two main things that can help improve your presentations.
Yeah, I think that's useful for not just scientists, I think for everybody.
It's useful for pitching, because I do a lot of pitching these days, because I'm interested in commercializing technology that I'm building. Pitching, teaching, everything.
Yes. Maybe that's for next episode of technology pitching. So I think we're about time, but I think for our trainees, so maybe I'll just ask one last reflective question. So now many trainees, maybe they are a bit unsure of their future path. I think we're all like that sometimes. So if they ask you, what's your biggest takeaway from your scientific journey so far, maybe life, personal, or science, what would you tell them?
I would say, trust your gut a lot. Maybe I'm just overly type B or something, but I think we need to get more comfortable making life decisions based on vibes, instead of just how it looks on paper. I know some people try and hack their PhD or hack their postdoc and find the exact PI that
I want this PI
because of his impact factor or her
all
years of experience,
fancy papers.
Or maybe they're a Nobel Prize winner or something. But what's really important is that you're going to feel that fire in your belly to be passionate about this work, no matter like what's going on around you, and that you are surrounded by good vibes that are going to support your mental health, your physical health, that you can maintain a work-life balance. And I think, it's that passion in you that's going to carry you. It's not, it's like, I don't know what else carries people, but like the passion. And if you are in a bad environment or a negative environment, just, or an unfunded environment or anything like that, you're not going to have that motivation to go into work every day and work on this thing.
Yeah. The PhD is like a marathon, right? It's like you have to be willing to do your day-to-day work. Right? You have to have something carry you on. Yeah. It's not just a passion and just like some fame or something that you can keep yourself working every day, right?
Yeah. And then I also want to just give a caveat to that. It's not like you're going to be motivated all the time. I think someone said this recently in a podcast on Mel Gibson. I love her. It was this self-made billionaire woman, and she said you need to expect. You need that motivation, but you also need to expect that you're going to have 1/3 of days be bad, and like where you just kind of feel crappy about yourself. 1/3 of the days are going to be meh. Okay, I'm just here. I'm just existing. And only about 1/3 of those days are you going to be like, yeah, this is a good day. I'm feeling good about my data. Expect 1/3 of your days to be bad, and then I think it'll help you manage your expectations about what you're doing.
Yeah, I think you have shared so many stories in your PhD, like not necessarily like always exciting, right? It's always some setbacks, and eventually you figure out, or you found a way and manage to put into your paper and becomes a good story, right? So it's like it's hard to be. I feel like whatever stage you're at, whatever occupation you're working on, it's hard to be happy every day. Right? It's hard to be pumped like every single second. Right?
And sometimes your bad data might actually be very insightful later.
Yeah,
if you're
that
lucky.
in mind.
If you're lucky. I'm waiting for that for myself in my postdoc.
Yeah. So maybe one last question. What are you thinking about your future career, or what are you anticipating like for now?
Yeah, my main goal is to commercialize what I'm developing in the Kaplan Lab. My PI, David, he is very supportive of commercialization opportunities and support, just helping out with that. I do see entrepreneurship, which is, bringing new things, new inventions and commercializing them. And I do see it as one of the best paths to material innovation and food innovation.
Especially what we're working on.
Right. Because I've talked to people who work, and people are like, oh, why don't you just go to work at Lycra and do, or work at Procter & Gamble or something. They might have some R&D departments that are working on these things, but they are so strict about, where the funds are being used and what constitutes a success versus not. And they are not patient, usually, the industry. Because it makes sense. They have to keep the business running. I
want their money back.
Yeah. I don't like doing R&D with the pressure of all this capitalism.
Yeah.
So I'm trying
from the investors, the board members.
I want to build this in an academic environment as long as possible, de-risk all 99% of my horrible ideas. And then when my 1% idea, successful idea comes out, I want to commercialize that into a new technology, and hopefully someone will buy it and I can exit, and then I can start something new.
I'm looking forward to that. Are you currently learning anything, because we have, few experiences on those things? Are you trying to, consult someone or learning something about, starting a business?
Oh yeah, because I'm a technical founder, and I do have a startup already on bio-based stretchy fibers. It's just still early in the R&D stage. It's a good way to get government money, from Department of Energy or Department of Defense.
Mm-hmm.
That's why we have the startup going already. It's called Good Vibes. But being a technical founder of Good Vibes, we don't have much training on the business aspect, and they don't expect us to. You don't need an MBA to start a new venture.
Mm.
You might want to partner with someone who is an MBA later, but you don't need that early on. So right now I do accelerator programs like VentureWell or iCore. The reason that I'm here today is because I was funded to come here for customer discovery by the Department of Energy. And so that
it's kind of business development
it's
stuff.
Business development, yeah. There's so many accelerator programs out there designed for technical founders who have their PhD or like, you know, they have an advanced technical degree, but they don't have that. It's so, you can learn, if you're a technical person, you can learn business stuff. It's hard to go the other way, to be a business person and learn the technicalities of
Could be
biology.
Intimidating a little bit.
But you can definitely learn the business stuff on the fly.
Yeah.
Yeah. So I'm learning that on the fly all the time. You know, I take little classes here and there, little accelerator programs here and there, and it's a lot of fun.
How do you feel about it? Like,
I feel like my technology is not ready to be commercialized, the technology I'm developing in the lab, but once I have that technology ready, I'm feeling very confident that I'm going to have so many people supporting me. I've had like hundreds of mentors out there right now. People who have told me, they're like, well, whenever you have something, like, just reach out, let me know, I'll get you with the right people. My brother, he's like, I know manufacturing. So my other brother, he's like, well, I know how to run business operations.
How did you meet those people in the field besides your relatives?
Yeah, besides my relatives. I just gave 2 relative examples. No, but there's other people like who are met in the accelerator programs who are like, hey, come shadow my angel investor meeting. Like, watch us, listen to our conversation and hear how we evaluate pitch decks. So I would meet, he was my mentor in a VentureWell program, a climate tech accelerator. And, you know, then he introduces me to folks who used to work at 3M or Dow or, you know, all kinds of people.
So those programs were not just based at one institute, right? It's
right.
Like more like just...
Yeah, no, you just like look them up online, like climate tech accelerators. You gotta be, I go to climate tech happy hours, I go to climate tech conferences, climate tech week at New York City. I reach out to people on email, hey, do you want to do a Zoom call?
Yeah.
Yeah, I mean, it's a lot of work, but it's kind of my hobby. I count business development as like it's my hobby. It's what I do for fun.
That's fair. Yeah. I think it's very interesting and quite useful for a lot of people developing cool techniques, right? It's good to know the other side, right? Like, how do you want to translate your research, right?
Right.
And it's good to network and meet more people, and you never know in the future.
You meet one influential person, they introduce you to 5 more.
Yeah.
And everyone in this space wants to help each other. No one's an antagonist. If you meet an antagonist, they've got something weird with them. You know, just move on.
Just had a bad day.
Yeah, like, everyone is rooting for you to develop something good for the world. The antagonists are the biomolecules themselves.
Well, the science at the end.
Yeah, that's every PhD student's antagonist. That should be the only antagonist in your PhD, I think. Your PI should not be, your advisor, your lab mates. No one should be an antagonist to each other.
I like that.
Only
thought.
the biomolecules.
Like that.
Only the RNA. Not
it's already hard enough.
Just
don't add more personal people factors.
And don't be an antagonist to yourself either. You do not need that. Only the biomolecules can antagonize.
Be kind to yourself. Yeah. Yeah. Well, I think that's it. Thank you so much, Blake.
This was so fun.
This is wonderful. I appreciate your honesty and a lot of insights you shared. And I'm sure this will resonate a lot with the trainees or even people outside science. I feel they will learn a lot from your experience. So thank you so much.
Happy to share my little, tiny.
Yeah, and also good luck
world.
with your future endeavors. I think
Thank you.
it's very exciting. I think you have a lot of foundations already built and just need some well-behaved biomolecules, and then
they need to behave better.
You
those
can just
little
skyrocket. Yeah. Thank you so
Thank
much.
you.
So we have a little gift for you.
Oh my gosh!
So this is a traditional Chinese bookmark. I know you're a book lover, so I hope this can decorate the book that you're reading a little bit.
Oh my gosh,
Yeah.
I love this. Wow, it's beautiful. Is that a temple?
I would say it's a palace or it's like a pagoda, but yeah, they all share some similar traits. So yeah, hope you can put that in your book that you're reading right now.
I admittedly I listen to a lot of audiobooks, but I do have some print books too.
Okay.
I love this.
I hope it can be used.
Oh, 100%.
Yeah. Okay.
Thank you so much, Henry.
Yes.
I look at it, break the fourth wall. Stay in school, kids.
Do you mind just do a clap to slate the shot for me? Like for the aligning the soundtrack and so do a big clap. Okay.
Is that loud enough?
Yes.